chelonia, etc. Third class is the hexaactinida, examples Euplectella and the Hylonema. This is the classification of the Porifera. Next point is the canal system in cyclone. The canal system of cyclone consists of many spongial, radial or vaguelated canals, excurrent canals, incurrent canals, and the last point is the significance of the canal system. Generally, we need first, there is no change in the syllabus. Generally, old syllabus, generally these topics, generally they have mentioned in the unit. Not a detail they have explained in the in these points. The next, the phylum Nidaria. Phylum Nidaria. The first point, general characters of the Nidaria. Into three classes. First class is the Hydrozoa. Examples of the Hydrozoa: the Hydra and the Obelia. Scyphozoa. Examples Aurelia. Third class is the Anthozoa. Example is the Maitreya. So this is the classification of data, and generally they have mentioned in the general characters at least 2 to 3 characters which are present in the class Hydrozoa, Scyphozoa, and the Anthozoa. Next, in this point, generally Nidaria. The Hydrozoa. These points generally they are present in the old syllabus, but generally these points generally they have deleted and they have additions of these points, importance and the types of the corals. The first point is the types of corals. The most of the corals generally belonging to the class. There are 2 classes are present, the Anthozoa and the class Hydrozoa. In the types of the corals, the hydrozoan corals and the anthozoan corals are present. Hydrozoan corals, the only 2 orders are present. Order Milioporina, Miliopora. Second order Stylisternia, for example Stylistes. Second is the Anthozoa corals. In the Anthozoan corals, there are 2 Corallian corals. Subclass Optecorallian corals belonging to the orders. Different orders are present. First order is the Stolonifera, for example Vipora. Order Ionasia, for example commonly known as the soft coral. Order Conarocaria, for example Heliopora, it is commonly known as the blue coral. It is commonly known as the sea fan or the horny corals. Second is the hexacorallian corals. In the hexacorallian corals, there are the different orders. The first order is the Antipatharia, for example Antiphatis. Order Madrioporaria, for example Phangia, Hevia, Esteporia, and the Menandria. These are the examples of the Madrioporia. And generally, they have the mentions in the list of the corals. In the list of the corals, the first corals is the Alsonium. It is commonly known as soft coral. Second, Tubipora, Organ, Pipora, Heliopora, blue coral, Gorgonia, sea fan, Phangia. It is commonly known as the mushroom coral. Next is the Favia, stony coral, and the Astrasia. It is commonly known as the star coral. Madrioporia as the stag horn coral. And the last coral is the Menandrina. It is commonly known as the brain coral. Generally, these are the list of the corals. Generally, they have included under these points. And next point is the importance of the corals. Importance of the corals. Generally, importance of the corals. These points. In these points, the corals are the shelters for the fishes. Generally, corals they are protect to the seashore from the erosion, and the corals generally they are produce the islands, coral islands from the habitats for the human beings and the land animals. For example, Maldive Islands, etc. Certain brilliantly to make ornaments like the mudra for the decorating houses, aquaria, or the rock gardens, etc. Are used for the constructing the building and the paving the roads. Generally, these corals they protect the seashore from the erosions. This is the importance of the corals. Next is the phylum Platyhelminthes. In the phylum Platyhelminthes, the general classification up to classes. Classification of the phylum Platyhelminthes. In the phylum Platyhelminthes, which are divided in three classes. First class is the Turbellaria, Trematoda, and the Cystoda. Turbellaria, examples of the Turbellaria, the Rugesia or the Convoluta. Examples of the Trematoda, Fasciola, Hepatica. Cystoda, for example, Tinea or the Echinococcus. So these are the examples of their classes. Next point is the life history of the Tinea solium. In the life cycles of the Tinea soliums, the first point is the morphology. In the morphological point of the Tinea solium, the three portions. Second part is the neck. Third part is the strobili. In the strobili, there are three proglottids are present: immature, mature, and the gravid proglottids. This is the morphological point. Next point is the life history. Life history, it consists of mainly the copulation and fertilization. Second point is the formation of the egg capsule. Third point, development. Fourth point, transmission to secondary host. Fifth point is the cysticercus or the bladder worm. And sixth point is the transmission to the primary host. And the next point in your syllabus, the parasitic adaptations of the tapeworm. In the parasitic adaptations of the tapeworm, it consists of mainly the 2 points generally they have mentioned: morphological adaptations and the physiological adaptations. Generally, in the unit second, there is a no one change generally they are present in the Nidaria. In the old syllabus, in the old syllabus, at this point, importance and the types of the corals. Only one change generally they are present into the phylum. The third, phylum Nemathelminthes. Phylum Nemathelminthes, the first point is the general characters and the classification of 2 classes. General character of the Nemathelminthes. Second point is the classification. In the classification of the phylum Nemathelminthes, the class Nematoda, for example, Ascaris, Wucheria, Trichinella. These are the examples. Second class is the Nematomorpha. Third class is the Rotifera, for example, Phyllodesis. Fourth class is the Chinorhyncha, for example, Echinodeus and the Trachydemus. And the fifth class is the Gastrota, for example, Macrodissus and the Chitonotus. So these are the classification of the Nematode. in these points and their examples. Second point is the life history of the ascaris. The life history of the ascaris, it consists of mainly the copulations and fertilization. Second point, zygote. Third point, early development. Fourth point, infection to the host. And the fifth point is the later development in the new host. And the last point is the parasitic adaptations of the ascaris. So these are the points generally they have mentioned. There is no change in the platyhelminthes. Generally in the old syllabus, these points generally they have present. And the next point is the unit phylum Arthropoda. In this phylum Arthropoda, generally in older syllabus, Arthropoda, the general characters and classification of they have deleted and they added these points Arthropoda as a vectors, mosquito, housefly, bedbug. In these arthropod vectors, the first point is the morphology, classification, and the mode of transmission of the diseases. The phylum Arthropoda, the first point is the general characters of the Arthropoda. Second point is the classification of the Arthropoda. The Arthropoda which divides into the 5 villa. Second class is the scorpion, spider, king crab. Class Myriapoda. Myria means many legs. For example, millipede, centipede. Fourth class Onychophora. Onychos means phora means bearing. For example, patus. Class Insecta. In Insecta, general characters of the Insecta, body is divided into 3 parts: head, thorax, and abdomen. Thorax, it bears the 2 pairs of the wings and 3 pairs of the walking legs. Example, cockroach, honeybee, and butterfly, grasshopper, etc. The arthropod as a vector. In this point generally, the new point generally in this syllabus, they add mosquito. In the mosquito, these are the points. First point is the classification of the mosquito. Second point is the morphology of the mosquito and the mode of the transmission of the diseases. Generally, biting mechanisms which are present into the diseases. The diseases generally, transmission of the diseases, malaria, yellow fever, dengue fever, filariasis or elephantiasis, influenza, and the dermatopia. So these are they have mentioned in the mosquito diseases. And the second point is the housefly. In the housefly, first point is the classification, second point is the morphology, and the third point is the mode of transmission of the diseases. Generally, the food contaminations and the diseases generally they are spread. The typhoid, dysentery, both bacillary or the amoebic, cholera, TB, leprosy, trichomonas, gonorrhea, and many helminthic diseases caused by Tinea solia, Ascaris lumbricoides, etc. These are the viral diseases of the poliomyelitis. So these are the next point is the metamorphosis in insects. In the metamorphosis of insects, which are divided into the different types. The first type is the ametabolous development or no metamorphosis, for example, Lepsima. Second, hemimetabolous or incomplete metamorphosis, for example, dragonfly. Holometabolous Holometabolous, the or complete metamorphosis, for example, housefly. Parametabolous or the gradual metamorphosis, for example, grasshopper. And the next point is the metamorphosis in insects, nervous and hormonal control of metamorphosis. In this control of the metamorphosis, there are different hormones are present. The first hormone, brain hormone; second hormone, prothoracotropic hormone; third, prothoracic gland hormone; and the fourth is the juvenile hormone. These topics generally they are included under the phylum Arthropoda. Only one change generally they are generally they add in this syllabus. Unit Annelida. General characters and classification up to classes. General characters of the Annelida. Second point is the classification. Phylum Annelida, which are divided into different classes. First class is the Polychaeta. Examples of the Polychaeta: Nereis. Class Oligochaeta, for example, earthworm, tubifex, etc. Third class Hirudinea, for example, the leech. Fourth class is the Archaeonellida, for example, Polygordius, Dinophilus, and the Protodrilla. These are the examples of these different classes. In this phylum Annelida, the addition of the syllabus, species of earthworm used for vermicomposting and the preparation of the vermicompost bed. The species of the earthworm, generally they are present. The first species is the Utrillus eugenie. Second species is the Icina foetida. Third species is the Pyronix sensibribacus. Family of these species is a Megascolicidae, and the Pantoscolex corenthyrus, deep soil dweller of this species. The Udigoster species, Polyperithema glonata. And in Maharashtra, the species like these are the Pontoscolex, Corythyrus, Perianix exactus, Perianix sensibiricus, Perithema elongate, Indrigaster, Idrulus eugenie, Icina foetida, and Perithema postum. By which earthworm species they compost a bed. This is not detailed. These are the next is the worm species for the vermicomposting. The Icinapoida red wiggler. These are the species of this red earthworm. They are used in the commercial composting, worm farming in a high environmental variations. And this is the manure of the vermicompost. They have mentioned in these points. These are the addition points in the new syllabus. Species of the earthworm. Next point is the phylum Mollusca. In the phylum Mollusca, the general characters and classification of two classes. In old gastropoda, these points generally present in the old syllabus. These points generally they have delete, and the addition points, scope of the pearl culture, species in the pearl culture. The first point, general characters of the mollusca. Do you know the characters? Different characters are present. Classification, which are divided into different classes. Class Arthropoda, there is a no shells are present. A means absence. Class Monoplacophora, deep sea shells, segmented animals they have present in that classes. Class Polyplacophora, for example, chiton. Class Gastropoda, in the gastropoda, examples snails and the sludge are present. Class Bivalvia, calms, mussels, oysters. Class Cephalopoda, examples of the cephalopoda, octopus, squid, cuttlefish, and the nautilus. And the class Scapopoda, tusk-like intelligent are present. These are examples. First point is the spawning pearl culture, and the second point is the species in which is used by for food, shell, and the pearl industry. Pearl is a white, highly shiny globular body found in the shells of the molluscan oyster. Pearl generally they are secreted by the mantle of the marine as well as freshwater bivalves, which is commonly called as the pearl oyster. The natural pearl formation, it is very slow processes. The oyster generally they are cultured for the industry. In the Japan, was the first pearl which is also used for the linga pearl. The types of pearls. In the types of pearls, there are three types: natural pearls, cultured pearls, and the artificially pearls. Natural pearls generally they are formed within the oyster or the mussels by either accidentally entrance of a solid or the accidental wound within the shell, muscles or the tissues. Pearl generally they are produced pearls. Generally, uh, next pearl is the cultured pearls. Cultured pearls they are produced for the human interference. When the pearls they are produced through the processes of the cultured pearl producing oysters and the mussels. Artificially pearls generally artificially pearls such a pearl generally deep made by the plastic, glass or the fish scales etc. With the artificially luster. The marine pearls, there are different species are present. The first species is the Pinctada fucata. Second is the Pinctada margigratifera. Third is the Pinctada maxima. These are the three species of the pearl oysters which are produced in the superior quality of the pearls. First is the Pinctada fucata. These are the commonly known as the Indian pearl oyster. Generally, it is formed known as the Pinctada martinezi in Japan. Both these species, which are known as the Pinctada fucata. Generally, it is the distributions of the Indian subcontinent, Japan, and the Persian Gulf. And the second, in India, these type of the pearls which are occur in the Gulf of the Kutch or the coast of the Gujarat and Gulf of the Mannar. Produced by this oyster were the famous of the linga pearls of the ornament oriental pearls. Second, Pinctada fucata, the pearls which are occurs into the intertidal area, the depth of this area 12 fathoms. It grows up to the size 9 centimeter and the maximum lifespan is about 7 years. The next second species is the Pinctada margifera. It is commonly known as the Linnaeus. This type of the oyster, it is commonly known distributed in India. It occurs in the intertidal region of the Andaman Nicobar Island. The black leaf pearl oyster shows about 7 meters in shale pearl oyster of the world. It is restricted in the sometimes they are seen in the Australia, New Guinea, China, Philippines, Thailand, Myanmar, Indonesia, etc. It also occurs in the Andaman and Nicobar Island. This is the morphological features of the Pinctada maxima, which are occurs into the low tide level about 45. This type of the species, it grows about 28 to 2 centimeter in diameter and weight up to 5.5 kg. See the species of the diagram generally they have shown as the Pinctada maxima. The next is the minor Pinctada species. Minor Pinctada species generally they are considered to be very important for the pearl productions, and these are the minor species of the oyster. Generally they have listed above the below the Pinctada albina, Carcharium. These species they are present into the shark bait. The pearls produced have strong incidence color, such a light yellow or the some colors it is a light brown or light green or the light pink. This is the minor species of the Pinctada albina, sugillata. This is the type of the species India, Gulf of the Mannar, Australia. And the next species is the Pinctada maculata. It is observed in the Australia. Pinctada timantisi, it is observed in India and Australia. Pinctada imbricata. These are species which are found into the Venezuela, Bermudas. This type of the pearls is a safe translucent color among the white, light color, pink or the yellow color. And the other species generally minor species, the winged oysters, window span oysters, sea muzze, giant carp, and the gastropod species. Heliatus species. It is a multicolor or the irregular pearls, especially which is a red and the green in a color. Gastropod species. Point in the freshwater pearls. The freshwater pearls they are produced by the freshwater muzes belonging to unit species. They are used the Hydrophilus sigilli of the Japan, Biscataria licata of China, and the species of the Lamelidians in India and South Asia. Lamelidians marginalis, these species they are used into the freshwater. There are two types, Gulf of the Mannar and the Gulf of the Kutch. These are the two mines generally they have mentioned into the pearl fisheries in India. So generally these are new points, the new additions, they are present into the mollusca. Portion in agriculture, there are two types are present, the marine water pearls and the freshwater pearls. The different species generally they have mentioned in these points. The next point is the phylum Echinodermata. The general characters and classification classes. First point is the general characters of the Echinodermata. Second point is the classification. In the phylum Echinodermata, which are divided into 5 classes. First class is the Asteroidea, class is the Aphiroidea, third class is the Echinoidea, fourth class is the holothuridia, and the class is the crinoidea. For example, starfish. Class of Iridia, brittle stars. And the class Echinoidea, example is the sea urchin. And the class Holothuridia, example is the sea cucumber. And the class Crinoidea, example is the feather star. So these are the classifications of the Echinodermata. So there is no change in this phylum Echinodermata. Second point is the water vascular system of the Asteroidea. These are the diagrammatically the system of the Asteroidea, and these are the parts of the this water vascular system. The first part is the madreporite, second is the stone canal, third point is the ringer canal, fourth point is the radial canals, fifth point is the Tiedemanns bodies, sixth point is the Polian vesicles, seventh point is the lateral canals, and the eighth point is the tube feet. So these are the points generally they have mentioned into the water vascular system in the Asteroidea. These are organic farming in old syllabus. Organic farming, the organic waste, types of the earthworm, preparation of the vermicompost bed, preparation of the vermi wash. These points generally they have neglected, and these points generally they have mentioned into the phylum Annelida. And organic farming, these points generally they becomes deleted, and the new points they add in this syllabus. Aquatic biology. In the aquatic biology, the points freshwater and the marine water habitat of the fishes. In the freshwater habitat, only one point types of the freshwater habitat. There are two types, lentic habitat and the lotic habitat. Lentic habitat means it is commonly known as a standing water system. It consists of ponds, lakes, swamps, etc. And the lotic habitat, it is commonly known as the running water system. It includes rivers, streams, springs, etc. And the second point is the characteristics of the freshwater habitat. So these points generally they have mentioned into the aquatic biology. In the freshwater habitat, different fishes generally they are present. And the B point, marine water habitat. In the marine water habitat, the characteristics of the marine water habitat. Characteristics of the marine water habitat. The physico-chemical characteristic features of the freshwater habitat and the marine water habitat. These points generally they have mentioned in these topic aquatic biology. Generally in BFY BSc students it is not a detail, but actually the only the what is the meaning of the freshwater habitat or the types of the freshwater habitat, characteristic features of the freshwater habitat, what is the meaning of the marine water habitat, characteristic features of the marine water habitat and their fishes. So this is the aquatic biology. Next, the thank you.
झालं का मॅडम? थँक यू.
जाधव सर. Thank you. Okay, sir.
हॅलो.
हॅलो.
हा. हॅलो.
हा हॅलो सर. हा येते.
कोणाला काही डाउट्स किंवा क्वेश्चन्स असतील तर विचारू शकताय. मॅडम जे टाइप्स ऑफ कोरल्स वगैरे आपण सांगितलं,
हम्म, हम्म.
त्यामध्ये इन्फॉर्मेशन देताना कोणती कोणती द्यायची म्हणजे सांगायची स्टुडंटसाठी?
इन्फॉर्मेशन म्हणजे त्याचं थोडं मॉर्फोलॉजिकल कॅरेक्टर्स किंवा जे पॉइंट्स दिलेत ना तुम्हाला लिस्ट दिलेली आहे.
हा.
इन्फॉर्मेशन म्हणजे काय की देर आर कोरलचे किती टाइप्स आहेत. क्लास टाइप्स ऑफ कोरल्स मध्ये हायड्रोझोन कोरल्स आहेत. तर हायड्रोझोन कोरल्स मध्ये देर आर डिफरंट ऑर्डर्स आर प्रेझेंट अँड देअर एक्झाम्पल्स.
ओके.
मिलिपोरिया, मिलिपोरा and anthrozoa are present. First is the octet corals. Now what is corals कोणते आहेत?
तुमच्या फोनवर
Generally different orders generally they are present. Generally these are the examples. Tribipora म्हणजे पाईप कोरल, त्याच्यावरती इन्फॉर्मेशन असेल किंवा मॉर्फोलॉजिकल कॅरेक्टरिस्टिक फीचर्स आहेत, these are the mentioned in these points.
ओके.
It is related to the practical and the theory.
ठीक आहे. थँक्यू मॅडम.
Generally, octacoralline corals and hexacoralline corals. So, in which these are the two types are present in anthozoan corals, and in which different orders are present, and their examples. So,
थँक्यू मॅडम.
ओके मॅडम. जातो सर, आवाज येतोय?
हा, आभार.
हॅलो,
हॅलो.
आवाज येतोय का?
हा, येतोय सर.
बोलते मॅडम.
थँक्यू मॅडम.
thank you so much.
एक मिनिट सर. मोरे सर.
हां, बरं. ते मॅडमला मला विचारायचं होतं.
हा हा, बोला ना, बोला.
पर्ल कल्चर मॅडमनी घेतलं की नाही,
हा.
त्याच्यामध्ये एवढे सगळे टाइप्स वगैरे घ्यायचेत काय BSc 1 लेवलला?
आपल्याला
BSc आवरला 2 स्पेसिसची जरी माहिती दिली तरी पुष्कळ झालं.
हा, ठीक आहे.
फक्त तुम्हाला तर हे माहित असावं म्हणून मी दिले त्या स्पेसिस. पण
कन्सेप्ट
हायलाइट केलं होतं की नाही? हा.
बरं, ठीक आहे. ओके.
2 species generally they have mentioned, and then it is a good quality for the vermicompost. Eudrilus eugenie and Echinus, these are the common species, and these common species generally they have also used for the vermicomposting. It is a proper by the earthworm species. या 2 species जे आहेत प्रॉपर स्पेसिस आहेत सर, त्या यूज केल्या तरी चालते. बाकीच्या स्पेसिस फक्त माहित असावं मुलांना किंवा प्राध्यापकांना म्हणून ते दिलेले आहेत. त्याची माहिती वगैरे आहे, म्हणजे मॉर्फोलॉजिकल कॅरेक्टर्स, किती लांबी, रुंदी, वजन वगैरे त्याचं कसं आहे किंवा काय, साधारण वर्मी कंपोस्ट बेड हे मध्ये मेन्शन करणं गरजेचं आहे. 2 स्पेसिस दिले तरी पुष्कळ झाल्या.
and detailed information about. Thank you very much, ma'am. So
Thank you,
we
sir.
proceed to the next session. For the next session, the resource person is Dr. R.B. More sir, zoology and fisheries in our institute, and his qualification is the MSc, MPhil, PhD, and his teaching experience is the more than 13 years, and 20 years is the research experience. He published the more than 15 research papers in international and national journals. He is also the member of the Institutional Animal Ethics Committee of Satara College of Pharmacy, Satara. Also the member of Board of Studies in Zoology, SGM College, Karad. Also the member of the Academic Council, YCS, Satara. And he also the recognized guide. Under his guidance, uh, uh, 1 students are already awarded, and he project. So Dr. R.B. More sir to start his lecture.
Okay, thank you, sir. Good afternoon to all. Before commencing my talk, I would like to thanks to the principal Dr. D.T. Zavudka and Vice Principal and the Chairman, Head of the Department of Zoology, G.Y. Unique and opportunity to share some insights of changes of the C1 in this faculty development program. Okay, so day before yesterday, we started this WP of the life science. Here, my talk is only related with the paper number 2, that is physiology. Okay. I am sharing my screen. Sadhu sir, is it visible?
Yes, sir. Yes, sir. Is it visible?
So, the talk is related with the paper physiology, the paper 2, VZT 101 of semester 1. And the unit number 4, it is the reproductive physiology. Okay. So, in this paper, there are 4 units. First unit, it is related with the physiology of digestion. Second unit is related with the physiology of respiration and excretion. And third unit is related with the cardiovascular system. Again, in that third unit, the ECG measurement and its significance. Right. The fourth unit is related to the reproductive physiology. So, in this physiology, the points which are given, it is the reproduction and endocrine glands and the physiology of male reproduction. It also includes the hormonal control of spermatogenesis. Okay. So, reproduction and endocrine glands under this title, the physiology of male reproduction and hormonal control of spermatogenesis, and the physiology of female reproduction, and again the hormonal control of the menstrual cycle. Here, the endocrine glands, which are only related, here we have to the reproduction. Okay. So, when we go to the physiology of the male reproduction, you must know you must have to teach the male reproductive system with these 2 points, that is the primary sex organs and the accessory secondary sex organ. Okay, so with these points, that is in the primary sex organ, the 2 structures are there. One is the scrotum and the testis, or the accessory or the secondary sex organ. Then the accessory glands, seminal vesicle, prostate and corpus, and the external genitia. Okay. Then the primary sex organs, that means the scrotum and the testes. So these are the 2 primary sex organs. First, you have to go for the definition of the reproduction. Then, in short, information regarding the scrotum, that is where it is located, its structure, and which muscles are attached to the scrotum, and the function of the scrotum. Again, information is related with the location, the structure, and it's a functional role. Okay, the major function of the testis, it is the gametogenic function or in the sperms by spermatogenesis, the sagittal section of the testis, that is the internal structure of the testis, and the inner the details along with the seminiferous tubules, then the coverings of the testis and the caput and the cauda epididymis, and how vasa differentiate is converted into the vasa differentiate. Okay. Accessory or the secondary sex organs, which includes the accessory ducts and the accessory gland. Accessory ducts, it includes the retitestis, vasa differentia, then epididymis, vasa differentia, ejaculate duct, and finally another duct is the urethra, which passes to the urinary organ. And the accessory gland, that is the three glands, seminal vesicle, prostate, and the Cowper's gland. So this is the information which is related with these retitestis, vasa differentia, and the epididymis. So these are the secondary sex organ helping in the milk production. Then vasa differentia. Its function is to carry the sperm from epididymis, the urethra, and the ejaculatory duct. So these are the pair of ducts which join with the vas deferens and the duct of the seminal vesicle. Okay, and it carries the seminal fluid and the spermatozoa to the urethra. Accessory glands include the three. It is the seminal vesicle. Seminal vesicle, it is a pair of small fibromuscular pouch. Okay, and the secretion of the seminal vesicle called as the seminal fluid contain the citric acid, fructose, fibrinogen, and the prostaglandin. Okay, so the fructose, it gives energy to the sperm.
हॅलो, आवाज गेला काय? हॅलो, मोरे सर. मोरे सर, आवाज गेला. हॅलो.
हा, एक सर मी बघतो जाऊन, काहीतरी आला असेल प्रॉब्लेम. मी बघतो एक.
हा, म्यूट झाले.
हा नाही, लेफ्ट झाले आणि म्यूटून लेफ्ट झाले. हा, बघतो.
म्यूटचं बटन दाबलं.
बघतो मी.
So back to the accessory gland. The accessory gland, that is the seminal vesicle, prostate, and the Cowper's glands, have secretion contribute in the semen formation. And the semen at the during only 3 to 4 ml in a single ejaculation, which consists 300 million sperm. And you know that despite the fact that only one sperm fertilizes the ovum, a large number of sperms are released to ensure the process of the fertilization. External genitalia, it includes the urethra and the penis. Urethra is the common passage for the flow of urine as well as the semen. So it is called as the urinogenital passage. So this urethra is divided into three regions. Prostatic urethra surrounded by the prostate, carries urine only. Membranous urethra located end of the prostate gland and at the base of the penis, and it carries both urine and the semen. And the canal urethra, which is situated in the penis and carries both urine as well as the urethra. Then the cylindrical, erectile, and the copulatory structure, which is actually used to transfer the semen into the female genital tract, that is into the vagina. Then we have to teach the histological structure of the testes in the transfer section. That is seminiferous tube, stages, that is the primary spermatocytes, secondary spermatocytes, etc. Then the Sertoli cells as well as the leading cells in the section. Finally, the major four components in the structure, that is tunica albuginea, then the seminiferous tubule, leading cell called as interstitial cells. And the role of the Sertoli cell, larger cells, which are tall conical cells extending between the basal membrane and basal membrane of the tubule to its lumen. Each cell consists of nucleus at the base, and which gives a bow to the developing spermatocyte. So here the spermatogonium is at the base. These are the spermatocyte, and this is the developing spermatid. So the function of the Sertoli cell provides the mechanical support and protection to the maturing sperm, in the nourishment to the developing sperm, as well as it helps in releasing the mature sperm in the lumen. So these are the functions of the Sertoli cell. And the leading cells or the interstitial cells, these are present in between the seminiferous tubules in the connective tissue. These are surrounded by the vessel, also known as the leading cells or the cells of the leading, which secrete the hormone testosterone, and it constitutes the endocrine part of the testis. This hormone is responsible for the development of the secondary sexual characters in men. Then the process of the spermatogenesis. So it is the process of formation of sperms. Spermatogenesis, it is sperms are developed from the haploid microscopic structure, that is the spermatogonia, and it is converted into the gamete known as the sperm or the spermatozoa. Okay, so sperms are developed from the diploid spermatogonia. The process of the spermatogonia, that is the testes, and each testes has the seminiferous tubule, which are lined by the pyroidal epithelium called as the germinal epithelium. And so from the germinal epithelium, the process of sperm formation takes place, while the Sertoli cells give nourishment to the sperm. So this is the process of the spermatogenesis. It is shown in these three stages. That is, the first stage is the multiplication stage, spermatogony, which is a diploid cell. It shows the multiplication. Then it enters into the growth phase, converted into the primary spermatocyte. It is again the diploid cell. The chromosome number, it remains 26. When the primary spermatocyte is get converted into it enters into the maturation phase, the meiosis takes place, and the secondary spermatocyte form, which is having the haploid number of chromosome, it is again divided. It is second meiotic division, again chromosome number 23, that is haploid remain constant. So from a single spermatogonium, the four spermatids are developed, which further produces the four sperms. Okay, so the process is called as the spermatogenesis. So in these three phases, multiplication phase, growth phase, and the maturation phase, sperm formation is completed. So the significance of the spermatogenesis include produces the haploid sperms, and in that process, the meiosis crossing over takes place, which produces the variation, and it proves the evolution. The point from the syllabus, it is hormonal regulation of the spermatogenesis. Okay, so the hypothalamus secretes the gonadotropin releasing hormone, which stimulates the secretion of FSH and LH, also called as the interstitial cell stimulating hormone ICSH, from the anterior lobe of the pituitary gland. This LH, it stimulates the interstitial cell, that is the living cells of the testes, to secrete androgens, principally the testosterone. So, the FSH stimulates Sertoli cells of the germinal epithelium to secrete the androgen binding protein. In short, it is written as ABP, androgen binding. Concentrate the testosterone in the seminiferous tubule. Finally, Sertoli cells also secrete the protein hormone, which suppresses the follicle. And this FSH, it acts directly on the spermatogonia to stimulate the sperm production. The mechanism of the hormonal regulation in the spermatogenesis is shown here. That is, from the hypothalamus, the gonadotropin releasing hormone, which further secretes the FSH releasing hormone, LH releasing hormone. It acts on the anterior lobe of the pituitary. So, this anterior lobe will secrete FSH and the LH. FSH further acts on the Sertoli cell, LH acts on the leading cell to secrete the testosterone. Sertoli cell, it secretes the APP, and further it activates the epithelium, and this germinal epithelium starts the process of sperm. And finally, the process of sperm formation takes place. Testosterone is not available, then LH is not secreted. And here, when the Sertoli cells are not activated by the FSH, the inhibin is not produced. So negative feedback, it is again goes to the anterior lobe of the pituitary, so FSH is not yet secreted. The stress is on. In absence of these hormone, interstitial cells undergo atrophy and it stops the functioning. So this testosterone, it is essential for the development, structural and functional maintenance of the male accessory reproduction. Genesis, the role of the FSH, then the Sertoli cells, how it stimulates, how the stimulation further takes place. These are the hormonal factors which stimulate the spermatogenesis. The role of the testosterone, role of role of the follicle stimulating hormone, role of the estrogen, controlling. So this growth hormone, it specifically promotes early division of the spermatogonia in its substance, as in a primary marked spermatogenesis is severely deficient or the absent in the dwarf cases, thus causing the infertility in the dwarf person. Point number 2 is related with the from your syllabus, that the physiology of the female reproduction and hormonal control of menstrual cycle. When we go to the female reproduction, we have to teach the female reproductive system. Okay, anatomy. When we see the anatomy, the female reproductive system, it consists of the three structures, that is two fallopian tubes, we show these two fallopian tubes. Here is the uterus and here is the vagina. Okay. So when you see the fallopian tube, it is divided into these three regions. So these three regions are shown here. In changing to the hormones. So these are the two structures in which the female reproductive system is shown here with the section, here with the entire figure, and this is the principal organ of the female reproductive system, and each ovary it contains 1 to 2 million primordial follicles. So these are the primordial follicles shown here, and it contains the primary oocytes that are called as the eggs. Further, these eggs that can supply that female enough follicles until that female reaches to her fourth or the fifth decade. When she becomes the 40 or 50 years of age. So these primordial follicles, these are arrested. All primordial follicles are arrested in the phase 1 of meiosis and the onset of the puberty. When the puberty gets started, the further development takes place. So this is the figure showing the section of the ovary, that two regions, that is the core portion, that is the medulla, and this is the cortex region. So in this cortex region, the developing follicles are seen, that is the primary follicle, then the secondary follicle, tertiary or the Raphane follicle. Here the process of the ovary, and the remaining follicle is converted into the endocrine structure called as the corpus luteum, and the pregnancy is not continued, it is converted into the corpus albicans. Various hormones, these are involved in the process of the ovulation. Okay, so in this figure, the various hormones are shown, that is the gonadotropin releasing hormone, which acts on the pituitary. Further, it releases the and the FSH. Egg or the ovum is released. It is further picked up by the fimbriae, it enters into the fallopian tube, where the fertilization takes place in this region. It is the for that 2-cell stage, 4th-cell stage, 8th-cell, and after that the implantation of that blastocyst takes place in the endometrium of the uterus. So there is hormonal control of the estrogen and the progesterone. So these are the principal hormones involving the reproduction of the female. That is the gonadotropin hormone, then follicle stimulating hormone. Third hormone, it is the estrogen. It is again the steroid hormone. Fourth one is the luteinizing hormone, LH, and the fifth one is the progesterone for maintaining the pregnancy. Then come to the reproductive cycle, that is the menstrual cycle. The fertile period of the female, it extends from the age of puberty, that is from 11 to 14 years, to the age of menopause, generally 42 years. So such fertile female, it shows two periodic cycles. The first cycle, it is related with the ovarian cycle, so the changes take place in the ovary, which occurs in the cortex region of the ovary, and the menstrual cycle that happens into the endometrium of the uterus. So two cycles are simultaneously carried out. One cycle in the ovary, another one in the uterus. So the phases of the menstrual cycles, these are under control of the hormones which are secreted during the different phases of the ovarian cycles. It is the periodic cycle, it is the rhythmic changes that occur once about every lunar month, that is 28 days in the organs, that is in ovary and the uterus. During the reproductive or fertile period from puberty, that is the age of 11 to 14 years, until the menopause, they include the ovarian cycle, the uterine cycle, it is also called as the endometrial cycle of the menstrual. This ovarian cycle in the cortex, and the luteals. Okay, so there are three phases of the ovarian cycles. So estrogen level, it stimulates the FSH secretion, and further new cycle is started. Now come to the menstrual cycle. This menstrual cycle, it occurs in the endometrium that occur in the ovarian cycle. It has a 4 phases. There are 4 phases of the ovarian cycle. That is the menstrual phase, then the regenerative phase, regeneration of the endometrium takes place, then the proliferative phase, and the secretory phase. Okay, and ovulation takes place at the middle of the cycle. So in this picture, the endometrial changes are shown here at the base. Then the pituitary and the ovarian hormones, how they are changed during the cycle of the follicular development takes place. It is shown here in the figure. So these first 4 days, these are related with menstruation. So the endometrium, it is washed out. And further, the regenerative process is started in the endometrium after the completion of the menses from day 4 to day 14. So slowly, the endometrial tissue, it is get developed. So at the same time, the FSH level, it is also get increased at the middle of the and the progesterone phase. And on the day 14, the LH surge is there. The process of ovulation takes place. The follicle develops the primary, secondary, tertiary follicle, Brachyan follicle is formed, and when day 14 comes, the ovulation takes place due to the surge of the LH. Okay. The empty follicle is get converted into the corpus luteum. If the pregnancy is not there, it is converted into the corpus albicans. So this is the entire cycle of the 28 days. It's the regenerative phase. It is called as the repair phase. Then the proliferative phase, also called as the follicular or post-menstrual estrogenic phase. Third phase is the secretory phase. Also, the endometrium, the changes in the glands, how they are spirally twisted. Glands are developed here in the endometrium. So the development takes place from the day 4 onward. The day 14, ovulation takes place, and further development continues. If no fertilization, no implantation, again the menses takes place. So this is the activity of the total 28 days. So these are the hormone level. Yeah, blood level of the estrogen, it shows increase at the when the day 14 comes here, and it remain constant. If no implantation, it again get decrease. So this is the development of the follicle. Development of the follicle. Here, the two hormones shown, that is the FSH and the the LH surge takes place at the day 14. Also, slowly the follicle FSH level, it is also increased up to the ovulation, and after that it is that slow down. So this is the by this way the hormones control the menstrual cycle as well as the these references you can use these references while teaching and correct the information of that topic. Thank you. Any doubts? No. Hello, Zadu sir.
Okay. Uh, so any doubt or queries to request to the all faculty to ask the resource person.
चिकमाळा माहित का जॉईन?
हा
चिकमाळ्यात जॉईन आहेत ना?
आहेत आहेत आहेत.
आहे तर चांगलं झाले.
तर किती डेपला शिकवायचं हे आपल्यावर अवलंबून आहे. BSc 1 ची मुलं आहेत काय त्यांना थोडंफार झालेलं आहे, परंतु त्या पेंडेमिक कंडिशन मध्ये काय झालंय,
हा, म्हणजे मला तेच विचारायचं होतं की BSc 1च्या मुलासाठी त्यातली डेप्थ किती ठेवायची.
थोडीशी डेप्थ आपल्याला वाढवायला लागणार, कारण गेल्यावर त्यांची लेक्चर झालेली नाही.
हा.
त्यामुळे
10 लेक्चरचेच आहे ना?
ऑनलाईन
हे
हो.
10 लेक्चर.
जाधव सर, आवाज मध्ये डिस्टर्ब होते सर. माझा पण तसंच झालं
हा
का
हा
आवाज?
हा मग हा तसं झालं. मला बी माझा नाव डिस्कनेक्ट होतोय.
काय नाही.
जाधव सर आवाज का होते असा?
इंटरनेट प्रॉब्लेम मुळे होत असेल मॅडम.
इंटरप्शन आहे सारखं.
हा
इंटरनेट कनेक्शन
त्यामुळे
अनस्टेबल
मध्ये
आहे
आधी
सर.
आवाज जातो.
हो.
ते आवाज बोला.
ओके
ओके,
ओके
ठीक आहे सर.
okay. Thank you very much, sir, for giving valuable information about the paper second and in this revised point reproductive physiology. So thank you very much, sir, for the giving valuable time and giving the most important information for the BSc first revised syllabi. In a few minutes, स्टार्ट केलाय दुबळ सरांना. बघू त्यांचा म्हणजे लवकर सुरू होत असेल तर एक 10 मिनिटात आपण पुढचं सेशन स्टार्ट करूया. अजून एकदा कॉल करून कॉन्टॅक्ट करू. एक 10 ते 15 मिनिटांनी आपण सुरुवात करूया. एक 15 मिनिटांनी सर जॉईन होतील. फोन झालेला आहे.
सर लोबळ सर जॉईन झाले.
हा झाले. एक 5-10 मिनिटात स्टार्ट करू सर.
बरं आहे.
लोबळ सर
नमस्ते.
नमस्ते सर.
खूप वाढवायला लागते ना.
हो बरं झालं आमचा वेळ काढला तुम्ही. काय आता वर लायब्ररी बसलाय.
हो लॅबमध्ये आता माझ्याकडे काय झालंय, मी जस्ट 2 मिनिटापूर्वी लॅबमध्ये FY बेसिस माझ्याकडे सगळं ऍडमिशनचं.
ऍडमिशन प्रोसेस
आणि
आहे का बऱ्यापैकी ऍडमिशन झाली का?
हो ऍडमिशन आहेत, चांगले आहेत, रिस्पॉन्स चांगले
आहेत ना.
होय.
हा
सर स्लाइड दिसते का?
हो दिसते.
काय झालं, मी खूप पळत गडबडीने स्लाइड्स बनवलेत सगळ्या. इफ एनीबडीला काय लॅकून असेल तर समजून घ्या.
मोरे सर सुरुवात करायची आपण सुरू करू.
करूया काय त्यांना पण आता गडबड आहे सगळी सगळ्यांचीच.
Ha.
ठीक आहे.
Okay. Welcome to all the faculty for the third session of the today's faculty development program. For the third session, resource person is working as an assistant professor from David College, Devadi, and his qualification is the MSc PhD, and he completed his PhD from the Aurangabad University, and he is also the head of the department of zoology in this college, and he also the recognized guide. Under his guidance, two students are working for the PhD. He published more than 10 research papers in international and national journals. The chairman of the Rayat Vigyan Prakalp in the Rayat Shikshan Sanstha. So, with this short introduction, I request to the today's resource person, Dr. M.J. Lubal sir, to start his lecture. Over to you,
Thank
sir.
you, Dr. Jadhav Ganesh. Thank you, Dr. Jadhav Ganesh. I am very much happy to discuss. I am not giving the lecture, but I want to discuss all the topics of the revised syllabus of BSc Part First Practical. First of all, I am very much thankful to Honorable Principal Dr. B.T. Jadhav Sahib, Honorable Vice Principal Dr. V.I. Deshpande sir, Head Department of Zoology Honorable Dr. More sir, माझे गुरुवर्य कारंजकर सर, आपण सर्व स्टाफ मेंबर्स zoology faculty. I have prepared the slides or the presentation in a very brief time. If there is lacuna, please help me. Can I start, sir, now?
Okay, sir. You can
Okay.
start.
Yes, sir.
Now, this is syllabus, or this is the part to give me to explain in details as well. First, practical is a study of following specimens: amoeba with amoeba, Eulina, Plasmodium, Paramecium with respect to their classification and locomotion. Second part of first practical is so much big. I will explain it later on. Third part is TS and LS of Psychon. Next part, life history of Tenia and Ascaris and their parasitic adaptations. Then third practical is preparation of hemine and homochromogen crystals. Next practical is study tour. Another one is measurement of blood pressure by sphygmomanometer. And last practical is recording of ECG. Now we will discuss about the practical first. Point number 1, classification and the locomotion. All we are zoologists aware about the this practical classification. First animal is amoeba. Amoeba is found in mud, freshwater ponds, streams, and ditches in which bacteria and decaying vegetations are very rich. The shape, as usual, it is very irregular shape. Body is covered by very thin and semi-permeable membranes called as plasma lemma. Protoplasm is divisible into an outer ectoplasm and inner endoplasm. The endoplasm is having a single nucleus, a large single contractile vacuole, and many food vacuoles. Holozoic nutrition and binary fission type of reproduction. And locomotion, amoeba, the locomotion of amoeba is called as amoeboid movement, and it is performed by the pseudopodia. Second animal is Euglena. Euglena is found in freshwater. It is freshwater flagellate. found abundantly in the ponds, ditches, pools, and slow-running streams. The body of Euglena is spindle-shaped, green-colored, measuring about 50 to 100 microns in length. The outer covering is called pellicle. The pellicle is marked by the spiral striations. The cytoplasm of Euglena is divisible into an outer ectoplasm and inner endoplasm, two-layered animal. The endoplasm contains nucleus, chloroplast, paramylum, and pyrenoids. The anterior end contains flask-shaped cytopharynx, or it is also called as gullet. The nutrition of Euglena is halophytic or saprophytic. Just below the cytopharynx, there lies a large contractile vacuole which is surrounded by the accessory vacuoles. Just above the contractile vacuoles, there is a stigmata or eye spot which serves as the photosensitive organ. It is nothing but the mass of the red hematochrome granules. I will go speedily. The locomotion of Euglena is due to the lashing movement of the flagellum, that is euglenoid movement. The movement by the flagella is called the euglenoid movement. Reproduction is usually by the binary fission. Third animal is Plasmodium. सर ह्याच्यामध्ये क्लासिफिकेशन पण आहे. मी थोडंसं शॉर्टमध्ये क्लासिफिकेशन घेतलंय. मी थोडासा जास्त जोर दिलेला आहे तो त्याच्या मॉर्फॉलॉजिकल पिक्युलरिटीज आणि लोकोमोशन वरती. नेक्स्ट एनिमल इज दी प्लाझ्मोडियम. प्लाझ्मोडियम इज malarial parasite. Life cycle of Plasmodium is completed and is completed in two hosts. First host is man and second host is female Anopheles mosquito. A sexual cycle is passed in man in two phases. First phase is liver schizogony and second phase is completed in red blood cells. This phase is called as erythrocytic schizogony. After liver schizogony, a stage called micrometacryptomerozoite invades the red blood cells and become rounded to form a young tropozoite. As the tropozoite grows in size, a central vacuole is developed. As a result of this, the nucleus is pushed to one side into the peripheral cytoplasm and forms a signet ring stage. These are different morphological peculiarities or the characters of the Plasmodium vivax. In sporozoa-like Plasmodium locomotion is gliding or metaboly by myonems. This is about the Plasmodium. Next animal is Paramecium. Paramecium is once again as like amoeba found in the mud, freshwater, ponds, ditches, in which organic matter and decaying vegetation is very rich. It is commonly known as slipper animalcule because the structure, the shape of the Paramecium is slipper-like. The body is slipper-shaped, covered by the pellicle and the cilia, and the locomotion of this particular Paramecium is by the cilia, also called as the ciliary locomotion, and a nutrition of this animal, that is Paramecium, is holozoic type of nutrition, and reproduction is asexual reproduction is by binary fission and sexual reproduction is by conjugation. Next animal is next practical is study of Porifera. In the Porifera, these are the general characters, and in phylum Porifera, there are different classes: Calcarea, Hexactinellida, Demospongia. We will see the general characters of the Porifera. These are strictly aquatic, freshwater, or marine animals. Multicellular body, but cells are present in the loose aggregation and tissues are not formed. These are diploblastic, two-layered. The outer layer is endoderm and inner layer is outer layer is ectoderm and inner layer is endoderm. In between these two layer, there is a gelatinous layer is called as mesoglea. The presence of ostia, osculum in current pores and excurrent pores, presence of canal system, spongin fibers, and spicules for the skeletons. Reproduction of poriferans is sexual as well as asexual. Phylum Porifera is divided into three classes, that is Calcarea, Hexactinellida, and Demospongia. The first animal is Cycon. Cycon is solitary colonial sponge found attached to the rocks and other substratum in the shallow sea water. The body of Cycon is slender, vase-like or cylindrical. The surface of the body of Cycon is perforated by the numerous pores called ostia. Ostia plays important role in the water canal system. Each cylinder is attached to the substratum by the base and opens to the exterior by an opening called as osculum. Osculum is fringed by the monaxon spicules, single arrow spicules, monaxon spicules. The body wall is made up of dermal epithelium. Skeleton of the cycon is mainly consist of calcareous spicules which projects out from the epithelium. Each cylinder has a central large cavity. That cavity is called as the spongocoel. Cell means cavity. The spongocoel opens outside through the osculum. Nutrition, respiration, excretion, all these functions are performed by the canal system. And reproduction of cycon is by both reproduction method, that is asexual as well as sexual. Next animal is Hylonemma. It is exclusively marine animal. It is found in the depth of 100 to 150 meters in the sea. Body shape is variable, usually rounded or oval in shape. The body looks like a structure of glass bowl with projecting tufts of the glass spicules. I have drawn in the diagram. The body is supported by spicules of 6 rays and small amphidiscs. The body is raised from the substratum by the stalk-like root tuft, which is twisted like a rope. Root tuft is made up of a bundle of very long anchoring root spicules. Root tufts also transverse the body as an axis or the columella and usually projects above the gastral cones. These are the morphological peculiarities of the Hylonemma. Next porifera animal is Euplectella. It is commonly called as Venus flower basket due to its beautiful, elegant, glassy shape. The animal measures about 15 to 30 centimeter in diameter. The body is long, rigidly curved and cylindrical. Body is composed of 4 to 6 rayed spicules forming three-dimensional networks with the parietal gaps. The spicules are joined together forming a network. It has knitted basket-shaped body, ostia and oscular sieve plates, hence it is called as Euplectella. Next phylum is Coelenterata. Coelenterata is again divided into different classes: Hydrozoa, Scyphozoa, and Anthozoa. We see the different characters of phylum Coelenterata. The coelenterans are aquatic animals. Some are freshwater, while some are marine water animals. They may be solitary, colonial, and sedentary, or free swimming. Am I audible, sir?
Yes, sir. Yes, sir.
They possess the coelenterate animal possess a cavity called a coelenteron, and these all the coelenterate animals are diploblastic, that having two layers, and also having non-cellular mesoglea. The animals are radially symmetrical and appear in two forms. First form is polyform and second form is medusiform. Reproduction, sexual as well as asexual type. Digestion of coelenterate of both type, extracellular digestion as well as intracellular digestion. Special types of cells are present in the coelenterate animals. That is, stinging cells are present. This phylum, as I have already told, the phylum is again divided into three classes: Hydrozoa, Scyphozoa, and Anthozoa. Hydrozoa, we will see the Obelia and Physalia. In Scyphozoa, we will see Aurelia, and in Anthozoa, we will see Tubophora. First is Obelia. These are the different morphological peculiarities of Obelia. Obelia is colonial marine sedentary coelenterate attached to seedbeds, shell, and the rocks. It lives in the colony. The colony of Obelia is made up of the branches and zooids. The colony is having as inner tubular and living part of the cynosars. The cynosars is surrounded by the tough and non-living layer. This layer is called as perisarc. The cynosarc is continuous with the zooids. The colony of Obelia is dimorphic. That means it shows two types of zooids. The zooids are the polyp, or also called as hydranths, and the blastostyles. The polyps or hydranths are nutritive in function and called as gastrozoids. The blastostyles are reproductive in function, so it is called as gonozoids. That means two types of zooids are present in the Obelia: gastrozoids and gonozoids. The perisarc is a yellowish or brown, tough, transparent layer. The perisarc is protective layer made up of the cuticle. This is about the Obelia. Second animal is Physalia. The Prostegues man of war is floating hydrozone colony consisting of four types of polyps. The Physalia contains four types of polyps. The nematophores functions as the float dactyl zooids, functions as the tentacles. Gastrozoids functions as the feeding zooids, and gonozoids produce gametes for the reproduction. The Prostegues man of war has two types of stinging tenocytes cells used to obtain the food with the help of the tentacles. Food sources usually consist of the food of Physalia is usually consist of fish fry and small adult fish as well as small crustaceans. Sensory cells of Physalia are numerous and located in the epidermis of the tentacle and the region around the mouth. Movement of the Prostegues man of war is done passively with the aid of its gas-filled float. The reproduction of Prostegues man of war is based on the colony of unisexual organisms, with each individual having specific gonozoids. Another animal is Aurelia. Aurelia has different morphological peculiarities. First one is it is exclusively marine and commonly known as jellyfish. Body of jellyfish is soft bell or umbrella shaped, perfectly transparent and bluish in color. The reddish or pinkish horseshoe shaped gonads are clearly visible from the surface. The circular body of jellyfish presents a concave oral or subumbrellal surface. From the center of subumbrellal surface hangs down a short mandibrum. At the free distal end, mandibrum is squarish mouth is present. From each corner of mouth hangs down a long tapering, much frilled, delicate oral arm. This is called as oral arm. Each oral arm has ventral ciliated groove leading into the mouth. The edges of arms are bisects bisects with the stinging cells called as nematocytes. The circular margin of umbrella is broken into 8 lobes by the notches. In each notch, there is a sense organ. That sense organ is called as tentaculocyst. Tentaculocyst. It is the sense organ. The free edge of umbrella is the base with closely set, delicate, hollow marginal tentacles. These are the different morphological peculiarities of jellyfish, or also called as Aurelia. Next one is Tibiophora. The first morphological peculiarity is the organ pipe coral can found in shallow waters of the West Pacific to the south of Japan. The habitat west to Africa's east coast and through the Red Sea. They prefer sheltered as on reef slopes. The organ pipe coral, the Tibophoria is also called as organ pipe coral, is unlike most coral due to a fact that it lacks colorful polyps, but it has a dark red colored skeleton composed of a thin tube that looks similar to the pipe organ. Next character is colonies of organ pipe coral polyps can form mound of up to the 50 centimeter in diameter. They also form symbiotic relationship with the zooxanthellae to obtain nutritional benefits. Another morphological peculiarity is, in addition to the polyps of the organ pipe coral, or also called as Tibiphoria, can use their tentacles to catch the planktons and other small microorganisms that happens to float by. With the help of these tentacles, it can float. The organ pipe coral is an important part of maintaining the balanced reef ecosystem that humans rely on for a number of reasons. It is currently considered near threatened on the IUCN red list. The Tibipora, or the organ, the pipe coral, is in the red list of the IUCN. It is threatened. Another one is Metridium. It is strictly marine, solitary or colonial, and are fixed to the substratum. These are only polyps. Medusoid stage is absent. In this polyp, medusoid stage is absent. Coelenteron is divided, and Misoglia contains cell as well as the fibers. Body is very short, cylindrical, and radially symmetrical. It is divided into pedal disc, column, and the oral disc. The pedal disc is attached to the substratum firmly, and the column forms a major part of the body. The column of Metridium is divided into an upper short thin-walled capticulum and lower thick-walled scapus. The capticulum and the scapus are separated by the prominent fold. That fold is called as collar. The oral disc is expanded and crowned with the several marginal tentacles around the mouth. It is dioecious animal and asexual reproduction by budding or also by the fragmentation. Next phylum is the Platyhelminthes. These are the general characters of the Platyhelminthes. First character is these animals are soft-bodied, dorsoventrally flattened, free-living, triploblastic, coelomate, and some are the endoparasite. For the first time in the animal kingdom, the head region, definite organ system like digestive, excretory, nervous, and reproductive systems is getting formed. Digestive system is incomplete due to the absence of the anus because it gets ready-made food material. Ingestion through the mouth or through the general body surface. Excretory system consists of phlegm cells. These are mostly bisexual, and fertilization is internal type. The phylum is divided into the following classes: Tubularia, Trematoda, and Cystoda. First animal is Taenia. Taenia is endoparasitic animal found in the intestine of the man. The body of Taenia is ribbon-like, differentiated into scolex, neck, and about 700 to 900 segments. Scolex contains 4 suckers and a rounded rostellum. The rostellum of the Taenia is crowned at its base by the double row of 2,832 hooks. Below the scolex, there is area of segmentation. That area is called as the neck. After the neck, a large number of immature, mature, and gravid proglotid or segments are present. Mature segments contains fully developed hermaphroditic genital organs. Male reproductive system consists of testes, vasa efferentia, vasa deferens, and the cirrus. Female reproductive system contains of a single bilobed ovary, oviduct, vitellaria, and the vagina. Ingestion of improperly cooked pork beef leads to the infection. जर आपण कच्चं किंवा कमी शिजलेलं बीफचं मटण जर खाल्लं, we will get infected. It causes various diseases in man like anemia, eosinophilia, diarrhea, hemorrhage, abdominal pains, nausea, etc. Next animal is Ascaris, male and female Ascaris. I think in syllabus there is mentioned male Ascaris as well as female Ascaris. Ascaris common endoparasite in small intestine of man. It is commonly known as roundworm. The body is elongated, cylindrical, and pointed at the both ends. The body of Ascaris is pointed at both the ends. Body is marked with the mid-dorsal, mid-ventral, and 2 lateral lines. There are 3 lines on the body. Mouth is situated at the anterior end. Posterior end of the male is ventrally curved, having cloacal aperture. Through the cloacal aperture, 2 spicules projects out. That spicules are called as pineal setae. Posterior end of female is bluntly pointed, having anus. The difference in male Ascaris, how to identify male Ascaris and female Ascaris, is depend upon that its posterior end. Excretory pore in male and female is situated at the anterior end, just below the mouth. Female genital aperture, it is also called as gonopore, which lies about 1/3 of the length of the body from the anterior end. Another differentiative character in male and female is male is smaller than the female. Anus in case of male opens in the cloacal aperture. Next phylum is Annelida. Annelida is divided into different classes like Polychaeta, Oligochaeta, Hirudania, and Archinidella. The different morphological peculiarities of Annelida are the annelidan animals are mostly aquatic, freshwater or marine. Some are terrestrial, burrowing, and some are found in the moist soil. The body is elongated, vermiform, bilaterally symmetrical. The animals are metamerically segmented. External segmentation is due to the transverse grooves, while the internal segmentation is due to the presence of the septa. The body wall is composed of the longitudinal as well as circular muscles and is covered by the cuticle. The body wall of Annelida is covered by the cuticle. These animals are triploblastic and coelomate animals. Respiration generally by the body surface. Circulatory system is well developed and circulatory system is of closed type. Blood is red in color due to hemoglobin which is dissolved in the plasma excretion by nephridia, locomotion by CT, or parapodia or by suckers. The sexes are separate or united. First animal is Aphrodite. It is also called as sea mouse. It is found in mostly found in USA. It is marine and burrowing form. Body of Aphrodite is very short and broad. The body shape, uh, the body of Aphrodite is oval in shape, dorsiventral flattened. Length of Aphrodite is 7.5 to 12.5 centimeter, and width is 30 to 35 segments. The ventral surface, it is the ventral surface is flat and segmented, consists of stiffed CT and forms a creeping sole of the body. Dorsal surface of the Aphrodite contains a soft notopodial long CT, forming a felt-like covering on the surface. Dorsal cirri are modified into plate-like structure called as electra. There are 15 pairs of electra. Head of sea mouse is situated anteriorly, having a median tentacle and a pair of lateral palps. There are greatly modified parapodia in Aphrodite. There are three types of CT present in the notopodia. The organism is identified on the basis of the CT. Soft CT, the soft CT forms the creeping sole of the ventral surface. Stiffed CT or the soft notopodial CT are modified into the felt-like covering of dorsal surface, and last CT are iridescent CT. First CT is soft CT, second stiffed, and third CTs are iridescent CTs. Anus is situated posteriorly on the dorsal surface. This is about the sea mouse. Next, annelidon is Nereis, also called as calm worms, can be found from the Alaska to the San Diego, as well as the Pacific coast of Russia in the mid to low intertidal zones. The Nereis can be found amongst mussels and branchials on fillings, sandy mud, and algal holdfasts. The calm worm, that means Nereis, is a segmented annelid worm. Its segments are wider than they are long. The prostomium has two antennae and a pair of pulp differentiated into two units. Pharynx of Nereis is delineated into two parts with a pair of stout jaws and conical teeth. There are usually uh, two pairs of eyes present most species of calmworms or Nereis. The color is greenish to greenish brown with slight blue tone. It can grow to a fairly large size at about 30 centimeter long and 1.2 centimeter wide. They usually eat algae and have large jaws with an irreversible pharynx used to tear their food from hard surfaces. Reproduction is done through the release of the body parts called as epitopes that makes their way to the surface in mating the swarms. After spawning, the male and female epitopes dies. Next animal is Peritima, earthworm. It is found in the moist soil. Body is long, cylindrical, and brown in color. Anterior end is pointed, while the posterior end is more or less blunt. Each segment of the body, except first and last segment, provided with the CT. The CT, next character is mouth is situated anteroventrally. This is differentiating character. Clitellum is present in the peritima. which is found in 14th, 15th, and 16th segment. 14th, 15th, and 16th segments gets united forms clitellum. Female genital pore is present mid-ventrally in the 14th segment. A pair of male genital pores lies ventrally in the 18th segment. Two pairs of genital papillae lies ventrolaterally on the 17th and 19th segment. The anus is present in the last segment. And sexual reproduction, the type of reproduction in Peritema postuma is sexual reproduction. Next animal is Hirudania. It is also called as Indian cattle leech, found in ponds, marshes, and the streams. It is ectoparasite. It sucks the blood from the mammal like the cattles. Number of body segments are 33. This is a specific number character. The number of body segments are 33. Each segment shows superficial divisions called as annuli. Presence of suckers for the locomotion and attachment. The suckers are present used for the locomotion as well as for the attachment. Indistinct head with 5 pairs of eyes situated dorsally in the first 5 segments. Animals are bisexual. The male genital pore is situated mid-ventrally in between the 10th segment. The female genital pore lies mid-ventrally on the 11th segment. The anus of leech or Hirudania lies mid-dorsally on the 26th segment. Only a well-developed posterior sucker is present. The anterior sucker is reduced or is nearly absent. Next phylum is Arthropoda. The phylum Arthropoda is divided into different 6 classes: Crustacea, Arachnida, Insecta, Myriapoda, Oncophora. There are different general characters of the Arthropoda. First one is they are aquatic, aerial, as well as terrestrial or burrowing and the arboreal in habit. Feeding habits: herbivorous, carnivorous, as well as omnivorous, or also called as parasitic. They are triploblastic animals, bilaterally cylindrical body and metamerically segmented. Generally, body of arthropod animals is divided into head, thorax, and abdomen, or also called as cephalothorax and abdomen, or head and trunk. The specific character of arthropoda is very important character. The legs are jointed. Digestive tract is complete, very simple and tubular and having various diameter. Exoskeleton with cutaneous cuticle. The circulatory system of arthropoda is open type. Cilia are entirely absent except Oncophora. The class Oncophora contains cilia. The presence of homocilia is a very distinctive character. Respiration direct or indirect with the gills, trachea, or by book lungs. Excretory organs include Malpighian tubules, coxal glands, or green glands. Sexes are separate. Some are hermaphrodite. Example, hermaphrodite is Balanus animals hermaphrodite. First animal from class Crustacea is Paleomon. Paleomon is also called as freshwater prawn. It remains at the bottom by the day and comes to the surface at the night time, and the prawn feeds on the algae, moss, small insects, and the debris. The prawn shows an elongated body which is spindle-like and shows a bilateral symmetry. The body of prawn is covered by the strong cuticular exoskeleton, and the body is divided into 2 parts: cephalothorax and abdomen. Cephalothorax has 13 segments. In that, out of 13 segments, First five appendages bearing segments constitutes head. The remaining appendages bearing segments from the thorax. The abdomen of prawn contains 6 pairs of appendages. These are called as the pleopods. This is the diagram of the freshwater prawn. Next animal from class Crustacea is Cancer, commonly called as rock crab. It is aquatic in habitat. The body is oval and flattened dorsoventrally. Eyes are stalked, and antennules are in the sockets of the carapace. 5 pairs of thoracic legs are well developed. The first pair of leg modified into the chelate, and this chelate is used for the offense and defense purpose. Abdomen is greatly reduced, folded ventral to thorax. Eggs are carried by the female with the help of abdominal biramus pleopods. Metamorphosis comprises zooia and megalopa larva. Next animal from arthropoda class Crustacea is Limulus. Marine animal. It is marine animal and is nocturnal. It is also called as horseshoe crab because its shape is being as horseshoe. It is also called as king crab or limulus. The body of limulus or horseshoe crab is divisible into cephalothorax, abdomen, and a caudal spine or a telson. The cephalothorax of limulus is covered by horseshoe-shaped carapace. It has a pair of median eyes and a pair of lateral eyes. On the ventral side of the cephalothorax, there is a mouth and 7 pairs of the appendages. Mesosoma contains 6 pairs of movable spines. It is the example of living fossil as they have not changed much from the last 350 to 400 million years. This is the very good example of living fossil. Next animal from class, first animal from class Archinida is Pelaminus. It is the example of, it is also once again the example of living fossil. It is nocturnal and strictly carnivorous. The body is dorsoventrally flat, divisible into prosoma, or it is also called as cephalothorax, and the opistosoma, it is also called as abdomen. Opistosoma is distinguished into 2 parts. First is mesosome and second is metasome. 6 pairs of appendages. Out of 6, first pair of chelicare, 1 pair is of pedipalp, and remaining 4 pairs are of walking legs. This animal is poisonous, having sting with poison gland, and the sting is present on the tip of the poison gland at the tip of last segment of metasome, or also called as that metasome is also called as abdomen. The respiration is by 4 pairs of book lungs. Sexes are separate. These animals are ovoviviparous. This is a very distinctive character. Ovoviviparous, egg-laying, as well as give direct birth to the young ones. Shows very ideal parental care. Vinchvacha birad pathivar. As example, parental care. Next animal is Scolopendra. This Scolopendra found under the stones or in the gaps or in the crevices. Body is elongated and dorsoventrally flattened with the numerous segments. Each segment bears a pair of legs. First pair of trunk appendages contains sharp paired claws connected to poison glands. This animal is carnivorous and feeds on the insects, spiders, worms, sludge, etc. And sexes are separate. Next animal is Jullus, or it is also called as millipede, commonly called as wireworm. It is herbivorous. This Jullus or millipede is found in dumpy and dark places under the stones or below the decaying leaves. When we disturb the millipede, usually rolls up into the tight coils. आता पावसाळ्यात दिवस आहेत, रस्त्याच्या कडनं जवळून साधारणतः कोकणाच्या एरियामध्ये, म्हणजे तुमच्या सातारामध्ये सुद्धा हे मिलीपेड्स रस्त्या कडेला डम्पी किंवा डार्क प्लेसेसमध्ये असू शकतात आणि ते डांबरी रोडच्या साईडला आपल्याला ते दिसून येतात. आपण जर त्याला टच केला साधारणतः त्याच्या बॉडीला, दे गेट्स coiled tightly. And the body of millipede is cylindrical, metamerically segmented and differentiated into head and trunk. Each segment each segment has a pair of legs. Such many pairs of legs are present. Stink glands present along the sides of the body, secreting noxious substance for the protection. This is the differentiative character. The stink glands are present and that secretes the noxious substances. Due to this secretion, they can get protected, and respiration is by the trachea. Next animal is well-known and common animal, Periplaneta americana. It is nocturnal. It hides during the daytime in the crevices, gaps, or in the holes of the kitchen, stores, rooms. Can I go next animal, sir? Because we all are well known about the Periplaneta americana. Sir, can I go next to next animal? Am I audible, Jadhav sir? Hello, Jadhav sir. Hello. हॅलो
हॅलो सर, बोला.
माझा आवाज येतोय सर?
हा, येतोय सर, येतो. कंटिन्यू. ब्रीफ मध्ये फक्त काय? हा,
एलिमेंट जरा जास्त आहेत.
ओके. हा,
येतोय सर, कंटिन्यू.
Okay. Next phylum is Arthropoda, and from class we have to study the apis. We are also well known about the apis. I will go to next animal or next phylum, that is mollusca. The mollusca is again divided into 6 different classes, one by one. This animal is chiton. All the zoology faculty is well known about the chiton. Another is dentalium. Third one is pila. Fourth one is it is also called as bivalve. Next one is logigo. Next animal from mollusca is sepia. This is octopus. सर मी जाऊ ना जरा फास्ट पुढे, कारण वेळ खूप व्हायला लागलेला आहे.
चालेल.
Okay, octopus. Then Echodermata. The phylum Echodermata is divided into 6 different classes: Astroridia, Echoinidia, Crinoidia, Ophiuroidea, Holothroidia. Just in this phylum, we have taken the single example. First is starfish, sea urchin, feather star, brittle star, and sea cucumber. First is starfish. We are well known about this starfish. Second is the Ophiura. It is also called as brittle star. This is Echinus from the phylum Echinodermata. This is Cucumaria. This is Holothroens. This is feather star, commonly called as feather star, also called as sea lily, generally found in all seas. Body consists of central part and arms. The all the arms are looks like the feathers, so called as feather star. Now this is practical number 2. LS of cycon, longitudinal section of the cycon. If we take the longitudinal section of cycon, we can see this type of structure. The body wall is diploblastic with an outer ectodermal and or dermal epithelium layer and inner gastric epithelial layer, or also called as endoderma. Non-cellular mesenchyme layer with the interspread monaxon and triaxon spicules is present in between two layers. The dermal epithelium is made by flat pinacocytes and is perforated by the ostia. The ostia leads into the incurrent canal, which leads through the prosopyle. Then it leads into the radial canals, which are lined with the choanocytes. The radial canals leads into the small excurrent canals, which are lined by the pinacocytes. The excurrent canal opens into the spongiocele, and the canal system of cycon is cyconoid type. This is about the LS of cycon, and this is this diagram is of TS, transverse section of the cycon. Second character is the second point is the gastric layer is made up of the mesenchyme with the interspread monaxon and triaxon spicules, and is lined internally with flat pinacocyte cells. Excurrent canals जसं एक्सप्लेशन वरचं होतं इलेस ऑफ सायकॉनचं, तसंच याच्यामध्ये मी घेतलेलं आहे. दिस इज नेक्स्ट प्रॅक्टिकल लाईफ हिस्ट्री स्टेजेस ऑफ द टेनिया. आता हे खूप मोठं आहे सर, मी थोडं पुढे जाऊ का?
ओके सर, ओके.
हे पॅरासायटिक ॲडॅप्टेशन्स थोडेसे इन शॉर्ट मी घेतलेले आहेत. 8-10 पॅरासायटिक त्याचे ॲडॅप्टेशन्स मी घेतलेले आहेत. नेक्स्ट ॲनिमल जो आहे आपला अस्कॅरिस. जसं त्याचे लाईफ हिस्ट्री ऑफ अस्कॅरिस ह्युमन आहे, पटल्या जड ते आहे सगळे. मी तुम्हाला ते ह्याची माझी PDF तुम्हाला पाठवून देतो सगळ्यांची. दीज आर दी पॅरासायटिक ॲडॅप्टेशन्स ऑफ अस्कॅरिस. This is practical number 3. As Ganesh Jadhav sir told me, please prefer about the practical number 3, Hemin crystals. This is aim of this practical is to prepare the Hemin crystals. Requirement for this practical is sterilized needles, cotton slides, cover slip, glacial acetic acid, and spirit lamp. The principle of this practical is hemoglobin gives red color to the blood. It is conjugated protein made up of non-protein pigment hematine and colorless protein globin. The hematine is made up of pigments. That pigments are called as porphyrin, which is combined with the iron. A hydrochloride of hematine forms the hemine. The procedure for this practical is take a drop of blood on the clean and dry slide. The slide should be clean and very dry. Allow drying and putting a drop of glacial acetic acid over it. After drying of blood drop, we can put a drop of glacial acetic acid and cover it with the cover slip, and then gently hit on the spirit lamp, and after the cooling, observe under the microscope. Under microscope, we can see the dark brown colored crystals or shining rhombic plates or prisms, and star-shaped clusters are seen. What is the application? For the application of Hemin crystal test, where we can apply. The Hemin crystal test is used in medico-legal works to differentiate fresh or dried blood marks from any other red coloring material. म्हणजे साधारणतः आपल्याला फॉरेन्सिक ज्या टेस्ट असतात त्याच्यामध्ये ह्या टेस्टचा वापर करता येतो. The shape of human crystals are species specific. It's very important character. The shape of human crystal is species specific. Preparation of next practical preparation of homochromogen crystals. The procedure of this practical is use the following procedure to perform the Tacoma test. आपण त्याला नाव या टेस्टला सुद्धा एक नाव आहे हिमोक्रोमोजन क्रिस्टल्स बनवण्याच्या टेस्टला. त्याचं नाव आहे Tacoma test. Place a small portion of suspected blood, scraped, teased apart, thread, concentrated extract on a microscope slide. Add a drop of Tacoma reagent and cover with a cover slip. Warm the slide on the hot plate. A characteristic reddish color will appear around the sample. This step is optional. Observe microscopically under the microscope for pink needle or rhomboid shaped crystals. या डायग्राम मध्ये क्रिस्टल्स दिलेले आहेत, त्याप्रमाणे क्रिस्टल्स तयार होतात. Actually, I have not done this practical. Next practical is measurement of blood pressure by sphygmomanometer. त्याच्यामध्ये मी what is blood pressure, how it is measured, स्पिग्मोमॅनोमीटरची ऍक्च्युअली टेस्ट कशी घेतली जाते, कसं आपण ब्लड प्रेशर मेजर करायचं आहे त्याची डायग्राम घेतलेली आहे आणि ऍक्च्युअल त्याची प्रोसेस त्याच्यामध्ये वेगळे मी थोडेसे पार्ट केलेले आहेत. वन बाय वन आपण थोडंसं बघूयात. द हार्ट सप्लायज द ऑर्गन्स अँड टिश्यूज ऑफ द बॉडी विथ ब्लड. With every beat, it pumps blood into the large blood vessels of circulatory system. As the blood moves around the body, it puts pressure on the walls of the vessels. Blood pressure readings are made up on two values. That values are systolic blood pressure as well as diastolic blood pressure. This is the sphygmomanometer. आता ॲक्च्युली प्रोसेस आपण आपल्या लॅबोरेटरीमध्ये खूप इझिली करत असतो, कारण तेवढं त्याच्यामध्ये महत्त्व आपण त्याला देत नाही, पण जेव्हा ॲक्च्युली पेशंटची टेस्ट घ्यायची असते त्यावेळी त्याला खूप महत्त्व असतं. मेजरिंग ब्लड प्रेशर ते सगळे सिस्टीम आहे. तर तुम्हाला माझ्याकडं स्पिग्मोमॅनोमीटर आहे, तो मी तुम्हाला दाखवतो. जस्ट अ मिनिट. सर दिसतंय जाधव सर?
नाही सर,
व्हिडिओ माझा ऑफ आहे का?
हा दिसत नाही सर, व्हिडिओ तुमचा
व्हिडिओ ऑफ आहे. ठीक आहे, माझ्याजवळ कोण नाही, मी आता जाईल लॅबमध्ये.
ॲपरेटस दिसले की?
हा, ॲपरेटस फक्त दाखवायचं होतं
हो
मला.
हो.
दिस इज कॉल्ड स्पिग्मोमॅनोमीटर. ते आपण आता दवाखान्यात गेलो की पहिल्यांदा डॉक्टर आपले किंवा आता नवीन प्रेशर मेजर करत असतात, इन्स्ट्रुमेंट्स आलेत त्याच्यामध्ये. ते थोडंसं आता जुनं व्हायला लागलेलं आहे. डिजिटल ब्लड प्रेशर मोजण्याचं एक युनिट पण आलं आहे, ॲपरेटस आलेला आहे. हा काल्फ वगैरे आहे, याचं आता नाव सगळ्यांना माहितीच असते. असे वेगवेगळे पार्ट्स आहेत. इयर आहे सगळं व्यवस्थित पाहतील. माझ्याजवळ कोण नाही, नाहीतर मी प्रॅक्टिकली ऍक्च्युली तुम्हाला करून दाखवत असतो. नेक्स्ट प्रॅक्टिकल, आय थिंक दिस इज लास्ट प्रॅक्टिकल रेकॉर्डिंग ऑफ द ECG. ECG मला शिकवायला म्हणजे प्रॅक्टिकल कारंजकर सरांनी माझ्याकडनं करून घेतलेलं आहे. आता कारंजकर सरांच्या पुढे हे प्रॅक्टिकल एक्सप्लेन करणं मला थोडंसं हे वाटतंय. कारण मी ज्यावेळी TYBSL होतो त्यावेळी ॲक्च्युली ECG तो ड्रम घ्यायचा, त्याच्यावर कागद लावायचा, त्याला गोळी द्यायची आणि तो फिरवून हार्टबीट ह्याचा घ्यायचा ECG आपल्या फ्रॉकचा. त्यामुळे ते खूप छान असं प्रॅक्टिकल होतं. हे जेवढं का मला छान असं एवढं काही एक्सप्लेन करता येणार नाही, पण मी जेवढं मला पॉईंट्स घ्यायचे होते प्रॅक्टिससाठी आपल्या, ते मी घेतलेले आहेत. थँक्यू सर.
Thank you, sir, for giving the valuable information and descriptive information about all the practical course. If any doubt or queries, please ask to the resource person. Okay.
हाँ ठीक है. आभार कोण म्हणते?
मानतो मी, sir. आता नाही.
Please.
It is my immense pleasure to propose vote of thanks of this session. So I'm very of the BSc first revised syllabi and
मॅडम माईक कोण मला कलेक्ट हो, पगमार मॅडम.
also to organize this faculty development program. I'm also thankful to the honorable principal, Dr. B.T. sir, who always supporting to organizing such a faculty development program and different programs. All the faculty members thankful to the patient listening to the all the sessions. Thank you very much. And with the permission of organizer, I declare that the today's faculty development.
हॅलो गोपाल
हा
सर.
सर.
हा थँक्यू.
आज थोडंसं गडबडीत बनवलं होतं, आता पालकांची पण वेटिंग आहे माझ्यासमोर.
असू द्या. ओके.
Nubal sir,
हा सर.
your presentation is as good as ready-made material for the journal.
Yes sir, I have written the journal.
Yeah, very nice.
I have written journal and also मी ते पब्लिश पण केलेलं आहे. पण थोडासा वेळ पण कमी होता, सरांचा फोन आला, म्हटले घ्या तुम्ही. जबाबदारी असते असल्यामुळे.
छान प्रेझेंटेशन झालं सर.
येस सर, याचा नमस्कार. थँक्यू. माझ्या मनासारखं झालं नाही तशी गडबड नाही. ओके, थँक्यू.
Okay.
Thank you.
Okay.
Thank you, all of you.
Thank
Thank you.
Thank you.
एंड करतो सर मीटिंग.
Okay.